Hello everyone, and welcome to my presentation. As it was introduced today, I'm going to talk to you about a scalable moment approximation method that is suitable for biochemical reaction networks of large sizes. So my main interest is describing the stochastic chemical kinetics. But to give you a little bit of a background on that, if you imagine a network of biochemical species that interact with each other via chemical reactions, that network is called the biochemical reaction network and is a model that is used to model several biological processes. For example, is it working? Okay. For example, signaling pathways or gene expression. As the simplest example, you can imagine a gene expression model where a DNA gets activated and then mRNA gets transcribed and translated into protein. Because of the stochastic nature of these chemical reactions, the evolution of the state of this process is also a stochastic process, meaning that at each time point you cannot deterministically determine what the state of the system will be, but instead this process can occupy a possible set of states with certain probabilities. And therefore at each time point you can associate a probability distribution over the state space of the system. So actually capturing this stochasticity is very important in understanding the underlying mechanisms of biological systems. So under certain conditions, the exact dynamics or the exact dynamics of this probability distribution can be described by the chemical master equation. However, which actually this chemical master equation is a set of ODEs that describes the probability of all possible states at each time point. But for most realistic systems, the size of the chemical master equation is very large, or even it's infinite, and therefore its numerical solution is intractable. So to remedy that, a range of approximations have been developed. For example, one can do stochastic simulations to directly generate realizations of this stochastic process and then calculate interesting features of the dynamics based on that. Or there is another group of approaches which try to truncate the infinite or very large system of chemical master equations into a finite subspace in which the numerical solution is actually tractable. But both of these type of approaches suffer from very high computational complexity and they become infeasible for realistic biochemical reaction networks. So this question is very valid to ask how we can capture stochasticity or heterogeneity for realistic biological processes. And in this regard, the method of moments, amongst other methods, have been developed that capture heterogeneity in terms of a few statistical moments of the probability distribution over the state space of this stochastic process. To be more precise, if you consider this probability distribution, you can write down the statistical moments, namely the means, the covariances, and higher order moments. And from the chemical master equation, you can directly derive a set of ordinary differential equations that describe the temporal evolutions of these statistical moments. Now, if for the moment we just look at the second order moments, so the covariances, we see that the number of covariances scales quadratically with the number of species, meaning that even the smallest set of moment equations that can give us any information about the heterogeneity, which is the second order moment equation, becomes quickly infeasible for large-scale chemical reaction networks. So we asked the question how we can improve this efficiency, and we asked whether all of these covariances are important or their contributions are significant for the solution of the moment equations. To show you a motivational example, here we're looking at the JAK-STAT signaling pathway as modeled by Bachman and colleagues. We took this pathway and simulated it with the full second-order moment equations. And over the course of the simulation, we looked at the pairwise correlation coefficients between each pair of species. And we saw that for more than 50% of pairs of species, the correlation never exceeds the magnitude of 0.1, meaning that perhaps a large portion of these covariances can be left out without harming the accuracy of the moment approximation method. Also to notice another interesting feature, we can look here and see that as soon as the reaction distance between the species in the network increases, the correlation coefficient drops very quickly, giving us the hint that perhaps we can use this network structure to identify which covariances are most relevant and have the highest contribution to the overall approximation quality. So with that, we had the idea of exploiting the network structure to come up with a scalable moment approximation method. But before we can define this model properly, we need to define a criteria for most relevant or most important covariances. And we noticed that in most applications, for example, in parameter estimation, one is mainly interested in the mean and the variances of species. Therefore, we defined the set of relevant covariances as those that influence the temporal evolution of the means and variances of species the most. And what does that mean? So we start by looking at the second-order moment equations, the full second-order moment equations as described here. These are, we look at only the ODEs for the means and for variances, meaning for cases where I and j are equal. And then we see which covariances show up in those equations. And based on that, we define a dependency matrix in which the ijth element is 1 if the species I is involved in the temporal evolution of the mean and variance of species j, and is 0 otherwise. But this is not yet good enough, because it means that every time we have to derive the full set of moment equations and then go through the equations and detect these. And that's not what we want. We want to have a structure-based scheme that can be very fast and efficient. So we were wondering whether we can find a link to the structure between the dependency matrix and the network structure. And by looking at the pattern of the moment equations, you can easily find this link, and it boils down to these two rules. That if you have a chemical reaction, the products of a reaction are directly dependent on the reactants and modifiers of the reaction, for example, the activators or inhibitors. And the reactants of a reaction are dependent on the other reactants and modifiers. So just by looking at the structure or looking at the stoichiometry matrix, you can write down the dependency matrix. And then the idea of the scalable moment approximation method is to only model those the covariances between the species that have a close dependency on each other. So now we can finally properly derive this. Again, I'm summarizing the second order moment equations here, where means and variances are written as functions of means, covariances, and higher order moments. By applying some moment closure technique, these higher order moments then are replaced by functions of means and covariances, and can be written in this closed form. In the scalable moment, scalable second order moment equation, which we call S2MA, we only choose a subset of moments that are denoted, it's denoted by I delta here. And only for those, we take directly the second order moment equations, and we replace the rest of the moments by 0. And how do we choose this set then? In the simplest case, we are interested only in the species that have a direct dependency on each other, meaning that if there is a non-zero, if the dependency matrix has a non-zero element between species I and j, then we model those covariances in S2MA, and for all other covariances, we replace them by 0. But one might want to go to higher degrees of this approximation to be able to capture long-range correlations and to be able to explain nonlinear dynamics that could be there in the reaction network. For that, one may want to also model the covariance between the species that influence each other, but via a delta number of species in between. And it turns out that those can also be very easily identified just by taking the deltath power of this dependency matrix. So no extra work here is needed. All right, with this, the S2MA is derived. Now we want to see how it performs and how much size reduction we can actually achieve by doing this reduction. So first we looked at a set of network motifs that occur commonly in biological networks. And we looked at a chain of monomolecular reactions, a two-dimensional grid of reactions, and a chain of biomolecular reactions with a hub. And for all of them, we see that the size of the first degree S2MA scales linearly with the size of the network or with the number of species, as opposed to the quadratic scaling of the full second-order moment equations. Now, if we don't want to restrict ourselves to just that, we can consider a general structure for network where we only allow for monomolecular reactions. In such a network, we can, for each species, we can define the degree of that node as the number of edges that are connected to it. And we can show that the size of this S2MA for such a network is proportional to the number of species times the average node degree in the network. Now, for biological systems, it's very often it's a very valid assumption to assume that there are no long-range correlations between long-range interactions between the species, and therefore one can assume that this average degree is independent of the size of this network, and therefore one can see that, again, the S2MA will have a linear scaling with the size of the network here. All right, but all of these are somehow simplistic or specific network structures. We were interested in seeing how this S2MA performs on realistic biochemical reaction networks. And to examine that, we looked at several pathway databases, and we simulated roughly 50 published pathways. We simulated them with 3 different degrees of scalable moment equations and also full second-order moment equations. And for comparison, we also show the results for the reaction rate equations here, which are a microscopic method and can only describe the mean behavior of the system without providing any information about stochasticity. It is very interesting here to note that the first degree S2MA has a linear scaling very similar to reaction rate equations for all of these realistic pathways, with the added value that it actually also describes the variances and some of the covariances. We see that as the degree of the method increases, the size of the second scalable moment equations also increase, but they all the time stay well below the size of the second order moment equations. So even if you go up to third degree, you still gain a lot of size reduction. So these are really promising results, but that's not all. This size reduction comes with the cost of losing some accuracy, because by leaving out some covariances and replacing them by 0, we of course introduce an approximation error. Now to study whether this approximation error is acceptable or not, we again looked at several chemical reaction networks, and we simulated them, and we compared the solution of the S2MA with the full second order moment equations. Here I'm showing you the results for one of these network motifs where we have bimolecular reactions. And for these 3 exemplary species, you can see that the results for the mean and variances obtained by scalable moment equations and full moment equations are practically indistinguishable. Now, if we go to a more complex system, again, if you look at the JAK-STAT signaling pathway, where we have also non-mass action kinetics, so non-linear propensities, we see that still for most of the time, the first degree S2MA does pretty well. But if one is not satisfied with the accuracy of that, then the approximation quality can be controlled and can be improved systematically by going to higher degrees of the scalable moment approximation. Yes, with that, I would like to come to my summary and outlook. So to sum up, I showed you how we can exploit the network structure and obtain a moment closure approximation that is suitable for large-scale biochemical reaction networks. And we saw that with that, we can actually gain substantial system size reduction, and in most cases, already with the first degree, we get a good approximation quality. But if that's not satisfactory, then we can control the approximation quality by the degree of the approximation method. Now, this work can be, of course, extended further. For example, the scaled moment approximation could be derived for third and higher order moments, or for modeling the variances that are, or for approximating the covariances that are not modeled by the S2MA, one can think of more sophisticated closure techniques rather than just replacing them by zero. For example, one can see if those can be explained as functions of partial correlations between the species. And lastly, it is very interesting to see how this S2MA does in real applications where capturing the heterogeneity is actually important. Two best examples in this regard are parameter estimation and model selection. With that, I would like to close and thank my collaborators, Fabian Theis and Jan Hasenauer, who are my supervisors for this work, and Michael Flossdorf, who is my current boss. And thank you all for your attention.
So thank you very much for your presentation. It was very interesting. I think the idea is very nice. I was wondering two things. One, if you are undertaking any analytical study of the error you commit in doing this, at least in some special case. And two, to what extent does moment closure blur the picture? Because it itself introduces an error that is not very clear.
Yeah, actually, those two questions are very important, and they're related because, so first of all, we could not do any analytical study on the error bounds because we always compare the approximation error of the scalable of S2MA to the full moment equations, and already for the full moment equations, there are no such error bounds because it's completely dependent on the moment closure technique that you choose. So that is a bottleneck actually, and it would be already very helpful for the moment equations themselves. But so far, I'm not aware of any analytical studies on that to get error bounds on either of these methods.
A couple questions here. Instead of zeroing out your higher order moments, can you either use low order approximators of those or incorporate an adaptive approach in your algorithm to control the error by adaptively including higher order methods, higher order moments when you need them? And the second question is, these correlations you're treating as static, but sometimes depending on the state of the system could vary, so from being important, and others could be important at other steady states of the system, and how do you hope to handle that?
Yep, those are very valid points. So first of all, I already forgot the first question. Can you just remind me?
Yeah, instead of zeroing out your higher order,
okay.
If you can
so, but you're asking about the higher order moments or the moments of the same order which are left out by the S2MA?
Both, I guess.
Both.
You can incorporate that activity.
Yeah, so we did not do a systematic study on that, but we looked a little bit whether it helps, for example, to still do this cutoff on the second order moments based on this structure, but include some higher order moments, and the results were not so promising. So, meaning that it would make more sense to stay within one order and try to describe that as best as possible before going to higher order moments. But of course, you can do you use a moment closure technique to approximate the higher order moments, and you can use something similar, so with the same assumption, so that your model is consistent, and with the same assumption you can see if you can then describe the moments that are left out instead of replacing them with zero. So that's absolutely true. And the second one, that is also very true that these correlations are completely parameter dependent, but in this approach we really wanted to just see what we can learn from from the structure of the network, and not having to do a computationally expensive simulation beforehand for different regions of parameter space and see which covariances are important. And of course, this method has its own disadvantages because of this. But I think if one wants to take into account the parameter dependency, then kind of have to take a different route.
Okay, I think we need to move on. Thank you again.